So in this DeBacco University video, I'm going to go into testing cannabis plant material for the all-important cannabinoids, looking at some of the analytical procedures that are done and the importance of doing this on cannabis plant material. All right, let's look at testing cannabis plant material for cannabinoids. Well, first off, here's the research article where a lot of the information is pulled from, so you're welcome to look at this in more detail if this is of great interest to you. So one reason why growers would test for cannabinoids, well, growers want to know how potent their plant material is because the composition of certain chemical compounds can largely impact the value and expected price paid for their produced end product. This is how you can start to see some different values here, different amounts, different costs, because it can relate to the potential of the concentration of cannabinoids and also potentially the terpene profile present. So why is testing for cannabinoids important just in general? You know, there's other testing that goes on. Keep in mind, you don't want to forget about microbial testing and mycotoxin testing and heavy metal testing as well. But why is cannabinoids important? Well, there's increasing evidence that the ratios of CBD relative to other cannabinoids, as well as endogenous chemicals such as terpenes and flavonoids, exhibit desirable entourage effect. So this is important as far as looking at the potential benefits to this plant material. The synergistic interactions between cannabinoids and other chemicals may be responsible for the medical benefits in the actual individual. This is why, in part, it's important to test for these, not just to claim the highest or particular numbers. So cannabis testing and the pharmaceutical testing practices is kind of like a merger between the two. So here we've talked about all the importance. Here we see a simplified version. Now we're looking at kind of what an analytical report can look like. And cannabis has cannabinoid percentages that are relatively high compared to other pharmaceutical compounds that may use bioanalytical trace analysis. Since it's common to dilute cannabis or hemp plant extracts by 100 or 1,000-fold or more to reduce the concentration of targeted cannabinoids to levels appropriate for the analytical quantitative determination. So that means cannabis, relatively speaking, to other pharmaceutical products is relatively concentrated, requiring it to be diluted down to be able to perform with some of the proper analytical tests. These analytical procedures need to be amended from current protocols to maximize the information content that is obtained to ensure the information you're getting as a consumer is accurate and representative of the actual plant material. Now, current popular technique when determining potency is HPLC, high pressure liquid chromatography. Using a photodiode array detector is commonly used in the cannabis industry because it's relatively easy and inexpensive to employ. And the different peaks will represent different compounds, and there's different columns that can be used. Column size is mobile phase buffer solution. A lot of information goes into those proper analytical techniques. Now the SIMLS technique, which is the selective ion monitoring liquid chromatography mass spec technique, can reduce the potential for unknown interferences due to co-illusion, which can cause go unnoticed with the HPLC techniques that are used. This is simply providing a little bit more of a precision way of analyzing those particular compounds. Now this liquid chromatography UV chromatography compared to this different analysis, we can see a little comparison here, and this is some of the data presented in that research article. This analysis of the sample shows much improved selectivity and absence of interfering chromatography components. So this is that, when we go back here, that selective ion monitoring liquid chromatography analysis. This is due to the increased selectivity techniques where only the protonated molecules of the targeted cannabinoids are monitored. So we're getting a higher degree of specificity. Here we're seeing this standard method right here, and then here we're seeing the selective ion method employed here. And this is the analysis of the same extract in comparison, where in A, the protonated molecules of the targeted cannabinoids, CBDA and CBD, along with THC and THCA, were monitored. We can see a greater degree of precision, hopefully a little bit more accurate results to get rid of some of what's called the data noise that could be employed. And lastly, just looking at HPLC compared to the two here, again, important to kind of like research and kind of have an idea of the testing methods available and what the lab that you're getting tested from, results from, might be utilizing. And in contrast to the HPLC method, if either the LC or the LC-MS, which is liquid chromatography mass spec analysis of the same sample is performed, the considerably higher selectivity of either of these techniques could facilitate qualitative identification of unknown compounds, as well as mitigate the potential for unexpected chemical interferences for the quantitative determination of the targeted compound. Great summary. Basically, it's more precise at looking at very specific compounds, which can be very beneficial when we're dealing with a plant material that can have a whole host of potential other chemicals or background noise there. We want to be very selective in the sense to ensure we're getting a quality result, a targeted result, and a very selective result. So while this might seem a little complicated, studying all of the analytical equipment, it's just part of you being an informed consumer for typical products that you may look at purchasing, or as a grower are looking at getting submitted to a lab for analysis.